Okay, let's get started. So hello everybody, I think by now you know me, this is Deepa. I will be presenting a research seminar today based on work that was actually started kind of here at the PopGen school about 2 years ago. So I'll explain that as we go along. Let me get going. You can see my screen, hear me talk.
Yeah.
Okay, all right. So I'm going to tell you today about some work we've done to understand the evolutionary impacts of bias mutation spectra, or to address the question of, there is mutation bias, but is it any good in molecular evolution in particular? And so hopefully many of you have already watched the short YouTube video, the primer that Lindy, my collaborator, has made for this particular project. But if not, I'll explain as we go along what I mean by these terms that you probably may not have heard before. So I think by now most of you are familiar with this idea of fitness landscapes, as flawed and problematic or narrow they might be, as Susanna sort of explained last time. But there's still a very useful heuristic, at least, to start thinking about how adaptation works. And you've heard Kavita talk about these, and other people during the meeting talk about this. So by now, I think everybody's familiar with what we mean by fitness landscapes. And we can think of adaptation sort of as a step-by-step process that is traversing this fitness landscape in genotype or phenotype space as a function. And fitness is a function of genotype or phenotype, depending on what we're mapping in our landscape, right? So you've again heard a lot about what determines which mutations are going to be leading to adaptation, or which are going to be observed during adaptation in different populations. So here, we may think of 3 different populations, and we notice that the purple allele has either been fixed or is at very high frequency in all of these populations. And we might infer that as the populations have adapted, potentially this is a function of this high allele frequency is a function of the population size as well as the fitness effect of each mutation. So on this kind of landscape, for example, we might imagine that if the black allele here is the wild type or the ancestor that we started with, maybe the purple allele is at high frequency because it has the maximum fitness benefit, and so a higher selection coefficient, assuming all populations are at a high size and so on, right? But there's another very important factor that tends to be forgotten in thinking of all of this, which is the impact of mutation bias. So if I tell you now that there is a bias in which mutational types are likely to happen, so there's a different probability of the types of mutations that can occur in a species, such that because of differences in DNA repair or specifics of proofreading, which Michael Lynch briefly went over earlier this morning, we might see that the purple allele is more likely to occur simply because of this underlying mutation bias, which is a feature of every organism's specific biochemistry, specific, the dynamics and the molecular basis of how mutations are occurring and which mutations are fixed and in what way, right? Sorry, which mutations are repaired, let me say, separate from the idea of fixation. And so mutation bias can have really large impacts also on which mutations are observed more often and what is their evolutionary fate is determined by the fixation bias then. So this is a raw material which comes into the population upon which selection or drift are going to act. So if this itself is strongly biased, it is going to change which mutations are likely to be fixed. And there has been some work.
Question. I didn't quite understand what you mean by mutation bias. Do you just mean mutation rate? Bias in substance?
Yeah, so I'll explain that a little bit more clearly with some examples later. But as just, I guess, the point, so you can define mutation bias in different ways. But the way that I'm going to discuss it today is primarily saying there is differential rate of mutations of different types, right? So for example, if you consider single nucleotide changes versus large mutations with indels, like large chunks of DNA disappearing or being inserted, these relative rates can be different, and the rates of these different types of mutations can differ across organisms. And so a mutation bias almost certainly exists in almost all organisms that we have measured we have any information about. But you can classify mutation bias in different ways. So I'll come to some examples, but it's basically differential rates of mutation, either in different parts of the genome or in different categories of mutations. Is that clear?
Yeah, thanks.
Okay, so here's an example of why mutation bias can be super important and why we need to think about it. So this is a study where they looked at different groups of birds, okay, which have adapted to high altitudes in different regions of the world. And so the ones you see in gray are the ones that have adapted to high altitudes. And this is a simplified phylogeny. This is not the full phylogeny of any of these groups. But the gray species and a sister clade has been highlighted for comparison, which has not adapted to high altitude, right? And in every time in the gray species, what you see is that there's a very, very high parallelism which mutation tends to be fixed. So the P, for example, is showing you the ancestral amino acid, and the letters here are showing you the new evolved or derived amino acid that's associated with this adaptation to high altitudes. And in many, many different groups of birds, this study shows that the same mutation or same set of mutation arises in each clade at the same positions. And earlier, we used to think that this is a feature or an outcome of very strong selection at specific sites, which are very important because of the important fitness benefit that they offer for, in this case, adaptation to high altitude. But what this study shows is that actually a large chunk of this parallelism can be explained by the fact that at CpG sites, which means C and G pairs that exist in genomes, the mutation rates are very high. And so all of these sites are cases where, because of a high mutation rate, the same mutation occurring again and again in different lineages is itself very high. And because that probability is high, then of course that's the variation which selection is going to sort from, right? So this is an example where mutation bias can drive parallel adaptation in different lineages. And there are other examples like this as well. Here's another interesting example from just a couple years ago. So here people have, the authors in this study tried to look at the drug resistance mutations that come up for specific cancers in patients, right? So the patients are put on some drug which is supposed to suppress the cancer and prevent further growth of the tumors, but they find that the tumor is growing anyway because it's resistant to these drugs. And so they did sequencing to ask what mutations are responsible for this drug resistance evolving as the cancer progresses. And they find that, you might simplistically say, based only on selection or based only on the selection coefficients, that you might be able to predict what is the allele frequency, right? So alleles which have, for example, a high selection coefficient, which are highly beneficial for the cancer to grow, you'd predict that they would be observed at high frequencies, right? Instead, what is found is the high frequency alleles are not necessarily the ones that provide the greatest benefit to the tumor. Instead, a large chunk of that allele frequency is explained by the fact that these red mutations, for example, are simply more likely to occur. So if you look at the underlying mutation bias in these cancer lineages, you find that the red mutations here, which are observed in high frequency, are more likely to occur baseline, forgetting their selection coefficients or their fitness effects, right? And so to be able to predict and understand drug resistance and combat it in this scenario, and this they've shown for multiple cancers, you need to understand the mutation bias, not only the selection, the selective effects of the new mutations, okay? So there are real, outcomes which are important to all of us to be able to understand mutation bias and how they impact evolutionary dynamics. So here's now a specific example that I'm going to refer to more often in my talk today. Mutation bias comes in many flavors. Like I said earlier, you can categorize mutations as the rate of transitions versus transversions, like I can explain here. You can categorize it as the rate of synonymous versus non-synonymous changes. You can categorize it as the rate of SNPs versus indels and larger mutations and so on, right? So today, like I said, I'm going to focus on transitions and transversions. So we have 4 bases, as you know. There are pyrimidines and purines. So mutations that take you from a purine to purine or a pyrimidine to pyrimidine are called transitions. It's shown in blue here. And all other types of mutations that sort of cut across these categories are transversions. So as you can see, the simple probability is that there should be many more transversion mutations. So there's no bias. You expect 2/3 of your mutations, or 66%, to be transversions. But what we actually observe is for example, if we look at all sequenced MA lines, mutation accumulation lines, which I hope you remember what they are, across different bacterial taxa, where you have a very accurate sense of what is the underlying mutation rate of different types of mutations, you find that the most bacterial species actually have a transition bias, right? So they're sampling many more transitions even before selection acts on them, all right? And transversions are relatively rare, even though the underlying baseline expectation is that transversions should be fairly common. So there's a strong bias in the mutation spectrum, okay? And we're saying that the mutation spectrum is biased. And the question I'm going to try and answer today is, why is mutation bias so common and how does it evolve, okay? So to address this, we collected random samples of mutations in bacteria with different mutation bias. So I'll talk today about work with E. coli wild type, which has a slight bias towards, or a fairly decent bias towards transitions, like most bacteria that we have understood or know something about. And the mutant we work with then is an E. coli strain, the wild type strain, where we've deleted the mutY gene. This is a DNA repair gene. And on deletion of this gene, this strain ends up sampling many more transitions, right? So we have now 2 strains which have very different mutation spectra or mutation biases. And we're asking what happens to the DFEs of these strains as they accumulate new mutations. So we do mutation accumulation experiments. So we have many lineages of the wild type. We allow them to undergo serial bottlenecks, and we will accumulate mutations in these lineages. Similarly, we do the same experiment with the mutY strain. And we sample different lineages at different time points where we expect to find single mutations, given the known mutation rate of each of these species or each of these strains. And then we sequence the genomes to identify strains with single mutations. So we had to sequence, for example, about 300 strains of each of these lineages to be able to identify 80 that had exactly 1 mutation with respect to the ancestor. And the reason we're interested in this is that that's the best measure of DFEs, right? That you have single mutations, then you're able to pin the DFE precisely to say, here is the DFE of single mutations, right? Because when you have multiple mutations that will accumulate in a lineage, you will have effects of epistasis, and we don't want to get into that, right? So then what we do is we identify these 80 strains for each of these ancestral strains, which are now separate from their ancestor by a single mutation. And we know exactly where that mutation is, what kind of mutation it is. And we measure the fitness effects of each of these mutations by measuring the growth rate. So we're just simply measuring the exponential growth phase of each strain in different environments. And we compare that to the ancestor to ask what is the relative fitness effect of each of these single mutations. Shraddha, question?
Hi, Deepa. Yes, I just wanted to ask whether when you say mutation, you mean a non-synonymous mutation or any kind of mutation?
Any mutation, including large indels. I mean, we are a little bit limited because of our sequencing. So if there are really big structural mutations, we are not able to identify them with high confidence, but those are relatively rare in E. coli in any case. So it's not, I think we got most of the mutations given the coverage that we had.
I see.
Okay, so as I told you before, if you look at what fraction of mutations are transitions versus transversions, we find that the wild type has about 45% transversions. Remember, the no bias situation is about 66% transversions. So there's a significant bias towards transversions in the wild type. And the mutY strain has 95% transversions, right? So it's sampling almost exclusively transversions. And what we find is the average fitness effect of transversions is about 7% beneficial on average. And so this means the S value is going to be 0.07. The mean fitness effect of transitions, on the other hand, is negative 4%, which means that there's a 4% reduction in growth rate, and the S value is going to be 0.4. So these are pretty large differences in transversions and transitions. And as a result, what happens is overall, we find many more beneficial mutations in the mutY strain. So 36% of mutations are beneficial in mutY compared to 28% in wild type. And this is being aggregated over different environments. Okay, so I'll show you the data in a little bit. But the point here is that we're seeing many more beneficial mutations in the mutY. So here's what those individual data look like for different environments. So just to clarify, we've done the DFE, or we've done the MA experiments in a rich medium, or LB for those who work with bacteria. And we're measuring the fitness of these single mutations now in many different environments, apart from LB, where we have a minimal minimal medium, where there's just the basic salts and everything that's required for growth. And then one of these carbon sources is being added, okay, which is noted here on the left. So these are simple stack bar plots showing you what is the relative fraction of beneficial, neutral, and deleterious mutations in each of these wild type and mutY strains for each of the environments. Okay, so let's start from these bottom 5 environments. What we see here is that there is no difference in the overall DFE between these 2 strains in these 5 environments. Okay, and that's kind of what you might expect as your null, given that starting off, we weren't sure that we would find any differences based on the different mutation spectra, right, in these 2 strains. So this is kind of saying, okay, this is what we sort of expected in a way, that there should be no difference. Then there are 2 environments where we find that, interestingly, the wild type is sampling more beneficial mutations. Okay, so if you look at this dark pink, compared to this compared to the dark green is bigger, that fraction of beneficial mutations. Okay, and interestingly, what we see is in 9 out of 16 environments, the wild type is sampling fewer beneficial mutations. Okay, and it's because of these overall effects. What we see then is that on average, the wild type is sampling more beneficial mutations, and it seems to be associated strongly with the fact that it samples fewer transversions, whereas the mutY strain is sampling many more transversions. Okay. Now, interestingly, if we ask what is the nature of pleiotropy with all these mutations, we find that mutY mutations are also beneficial in many more environments. Okay, so let me walk you through this. We can to think about pleiotropy. So pleiotropy is basically situations where a single mutation has effects on fitness in multiple environments. Okay, now these effects may be synergistic or beneficial. So, for example, if you're plotting in this abstract cartoon here, the fitness in environment 1 on the X and fitness in environment 2 on the Y. So the center point here is going to be a case where there is no effect on fitness in any environment for a given mutation. Any dots that fall, or any mutants that fall in this quadrant, are beneficially synergistic mutants. What that means is they're beneficial in both environment 1 and environment 2, so they increase fitness. This is the opposite case where there's deleterious synergistic pleiotropy. That means these are deleterious mutations in multiple environments. And these 2 are cases where you might expect to see trade-offs due to these single mutations, where a mutation is beneficial in one environment, but deleterious in the other environment, and vice versa. Okay, so one question in understanding adaptation is how many of your mutations are showing each of these kinds of outcomes. And so we asked in the wild type scenario, where this is kind of what the DFE looks like. The green is showing the wild type and the pink is the mutator, the mutY strain. You find that about 12% mutations show synergistic beneficial pleiotropy, 40% show no change in fitness in multiple environments. So fitness changes in only a single environment, and so on. Okay. Now, interestingly, in the mutY strain, what we find is that the incidence of beneficial synergistic pleiotropy has increased quite a bit. So more than 20% mutations now in the mutY strain are beneficial they fall in this quadrant. So that means they're beneficial in multiple environments. And now, interestingly, what we do is we asked whether this difference is caused simply because the DFE is more shifted towards the right or towards the beneficial side in the mutY strain. So what we did is we did simulations where to the wild type DFE, we just to all the measured S values that we had for any given environment, we just added an equivalent amount to shift the wild type DFE towards the beneficial side, okay? So that its average S effect is matching what we see for the mutY strain. Now, if you do that simple sort of shift, what you see is automatically the beneficial synergistic pleiotropy increases, which kind of makes intuitively sense, right? That basically what you're doing is you're making more mutations beneficial in more environments. And so just by that shift alone, you're going to observe more beneficial synergistic pleiotropy. In contrast, if we now shift the wild type DFE in the opposite direction, we make it more deleterious by the same amount as we did here. we find that this fraction of beneficial synergistic pleiotropy goes down. Okay. Now, what this means then is ultimately that there are any beneficial mutation is beneficial in many more environments in the mutY strain. So here what we're plotting is we're asking all the beneficial mutations that are observed in the mutY or in the wild type strain. How many environments are those same mutations beneficial in out of the 16 environments that we tested, right? And what we find is on average, just above 5 environments show a beneficial effect. A mutation in the mutY strain shows a beneficial effect in just over 5 environments, whereas that number is as low as maybe 2 or so for the wild type strain. And in complete opposition to that, the number of environments in which a deleterious mutation is deleterious is higher in the wild type compared to the mutY strain, right? So what this means is, going forward, what we would predict is if this DFV dictates what happens during adaptation in different environments, okay, what's going to happen is the wild type is less likely to adapt to different environments as fast as this mutY strain should, because it's more likely to sample beneficial mutations in multiple environments. Okay, so there are important consequences then to the potential impact of this mutation spectrum that I mentioned to you. Now, it's also possible that the differences in DFE we see are not actually explained by the fact of the mutation bias, but maybe it's explained by the fact of initial fitness being different between the wild type and the mutY strain, or it is explained by epistasis, right? So to test this, we asked whether the growth rate or the fitness of these two different strains is itself different, the ancestors is different across different environments consistently, and we find that it's not the case. So if in general, the wild type is beneficial, has some advantage because we haven't deleted this particular gene in the wild type, we would expect that in general, the wild type would have higher fitness than the mutator or the mutY strain in most of these environments, and we don't see that. Okay. Similarly, we took a subset of about 20 mutations that were observed in the wild type strain, and we also made them in the mutY background, so that now we have the identical mutations occurring in a wild type and a mutY background, genomic background, to test for any epistasis, right? And we find that the difference between the fitness of these two kinds of strains that we generate is 0 in most cases, in most environments, right? So what that means is the fitness effect of a mutation in our set of mutations is not dependent, is not changing based on epistasis with the genetic background, right? So regardless of what strain background that mutation is in, its selective effect remains more or less consistent. Okay, so that means that we can't explain the differences in DFVs that we observe based on epistasis or based on initial fitness. And so we're left with this idea then that it's the mutation spectrum and the differences in fraction of transversions and transitions sampled, which is responsible for the observed fitness DFE differences. Okay. Now, this is a bit of a funny thing then to have, because there's no reason to expect why transversions in general should be more beneficial than transitions, because the fitness effect of any mutation is going to be determined much more by its local context. What amino acid, is it a synonymous, non-synonymous change? Where is it in the amino acid? What is the protein doing, et cetera, right? And so it seems very strange to say that it's transversions versus transitions which explain what is going on. There's a question, Nikhil?
Yes, maybe you'll talk about it in a couple of slides, but I was just losing patience. But besides this possibility that there can be difference in the mutation rates, it's also possible that these two strains, wild type and mutY, they belong to different points in the landscape, and somehow the topology of the landscape dictates which are the next mutations will be, are they going to be beneficial or not? So that can be an additional.
Yes, I'll come to that. Yeah.
Okay.
Yeah, but the bigger question is, your intuition is right that they're occupying different spaces in the landscape, but why should it be better to be in one spot, right? Like, why is the mutY strain in a nicer spot compared to the wild type? Okay. And so I'll come to that. Yeah. Okay. Any other questions so far? If anything was not clear. Okay, good. So we had to then ask this more general question: when are shifts in mutation bias generally beneficial? Are they generally beneficial at all? Or is our case, just some funny thing, there's some odd coincidence that we are finding out? And so this is actually the stage we were at, more or less, not exactly. We were still struggling to understand all of this 2 years ago when we had data for all of this. And we were sitting in this PubGen school and listening to lectures. And I started talking to Lindy to ask, say, hey, what do you think? Like, can we model this maybe? Can theory help us understand this a bit better? Because there's a limit to experiments, how much we can do with experiments. And all of the work I showed you is a ton of work already for anybody to do. And so we were thinking about this in the few days of the PubGen school that Lindy was here. And that developed into a very nice collaboration where she did a lot of simulation work and thinking about how to build analytical frameworks to address this question in a more generalized way, right? So going beyond our specific strains and our specific sets of mutation biases and what we can do with our experimental data to ask a more general question that are there situations or under what conditions will shifts in mutation bias, like what we generated when we deleted the mutY gene, are they often beneficial? And under what conditions will they be beneficial, right? And so what I'm telling you about these simulations is all work done by Lindy for this project. And so she conducted simulations of adaptive walks on random fitness landscapes. And so she's constructing these sort of generic landscapes with an NK model, which I think you've been introduced to already earlier in previous talks. And let me give you an example of what this simulation looks like, right? So we're just zooming in into some piece of this landscape. Let's imagine this is where our ancestor is at the starting point, right? So it's at some sort of lower level of fitness compared to the peaks of fitness that it could reach. We ask in this particular landscape, so we start with the ancestor with a random genotype, okay? So let's imagine we start with the 8 base pair genotype. We can't do real genomes because that's too much computationally, and we don't need to in some sense. So we do, let's say, a 8 base pair genotype sequence for this ancestor. It's randomly chosen. And we have an underlying fitness landscape, which has been randomly again specified, okay, in the simulation itself. So based on what the genotype is and based on the random landscape that has been generated, we have already a fitness value for this starting ancestor, right? And that's why we know where it sits on this landscape. Now, what we can ask is we can ask if if this ancestor takes a one-step mutation, okay, a single genotype, single base pair changes, it acquires a mutation. Here is a circle of sort of genotypes that it could sample, right? And keep in mind, I'm squishing a 3D or multidimensional landscape into a 2D picture here, but bear with me, right? So it will be something along the line of some sort of sphere or some sort of circle around this, right? So these are all the possible single-step mutations that could occur. Okay, now a bunch of these are going to be deleterious, right? So again, based on the underlying landscape. And so we want to discard these because we are looking at adaptive walks, right? So these are not random walks, these are adaptive walks. So we discard the deleterious mutations, but from the available available beneficial mutations, we randomly sample one, okay, according to the mutation bias, which we have already encoded. Correct. So if, for example, transversions are more likely to happen in this ancestor, then transversions are more likely to be chosen of the beneficial mutations that are sampled. Okay, so let's imagine this mutation was sampled and that constitutes one step in this adaptive walk. Then let's imagine we do the next step where again, some mutations are sampled, one is chosen, and so on, right? So there's this step-by-step process through which the population is moving in this landscape. And we're doing these simulations in a weak selection, strong selection, weak mutation regime. So any mutation that comes in has that is chosen is immediately fixed in the population. Okay, so we're pretending that it's a super large population. It gets things get fixed very quickly. All right. Okay, so then we do, so this is using an NK model, which where there are n sites in your genotype and k of them can interact to affect fitness. So there is some epistasis we build into it, even though in our experiments, we don't have any epistasis because we look at single mutation parameters. Okay, and we do 500 such walks on 500 randomly generated landscapes. And what we can do is during the walks, we can then simulate what would happen if there is a shift in the mutation bias, right? So we can start a wild type ancestor evolving in the landscape, and then we can say, okay, what happens if we now allow the evolution of a mutation bias, such as mutY, Okay, and that is an instantaneous shift. And then the next steps are taken in accordance with the new mutation bias. Okay, so in general, in adaptive walks, yes,
need to understand this. So, so the usual adaptive walk is performed starting from some initial fitness. And then when you say you introduce mutation bias, what does it mean? Meaning starting with a different initial condition, or I still don't get the point.
Yeah, so we start with the same ancestor, whatever we chose. So the ancestral condition is chosen again, as you'll see in the results. I think later it will become a little bit clearer, but you can have your starting condition be a bunch of different biases, right? So you can start with, as an example, I'm going to say what happens with the wild type starting bias. And at a particular time point during the adaptive walk, so at different time points during the adaptive walk, we can simulate a shift to a different mutation bias. And so what happens is basically...
So I think that's what I'm missing still. So like when you say you shifted, I mean, the mutation bias occurs, what does it mean in terms of the model? So what I'm thinking about is, like, for example, like this, suppose I have just a three sites, so 0, 0, 0, but that's my wild type. And it has these three neighbors. So let's say 0 becomes 1, I get 1, 0, 0, and then, perform the walk by changing one site at a time. So that's your the first thing. And then now you say that you want to repeat the simulation with a mutation bias. So where is the change happening in that usual standard?
Yeah, so I think the model you're thinking about will not work exactly because it's the binary, like 0-1 system. I guess you could make it a 0-1 system for two types of mutations.
Yeah, you can generalize. You can have all four.
Yeah. So basically what we're doing is so when you're sampling in a 0-1 model, you're saying there's a uniform distribution of sampling 0 to 0 or 0 to 1, right?
No. Because you will tend to, because it's adaptive walk, you will generally go to the one which has a higher fitness.
But that step is next step, right? In your initial sampling itself, your initial probability is the same for both kinds of mutations happening. Correct?
Yeah.
So
I mean, there's no mutation, right? I mean, mutation really... okay, so I think what you mean is that the mutation rate is same for all the three of them.
Correct.
So whenever I choose something preferentially according to fitness, but also I'm assuming, yes, I think, yeah, you're right. The mutation rate is assumed to be same. So now you're saying mutation bias means to which side changes. I go to, mutation rate is also non-uniform.
Yes.
I see.
And so it's not non-uniform across sites. It's non-uniform across which type of mutation is going to occur. So
maybe a couple of classes in which one of them I go to more preferentially. Is that the sort of thing?
Yeah, exactly. Yeah.
Okay, fine.
So if, for example, 0 and 1 are two kinds of types of mutations, we're basically saying now if initially the wild type has no bias, right? A 0 site is just as likely to mutate to, let's say, a 1 site or a number 2 site, right? A value of 1 or 2. But in our biased situation, let's say 0 to 1 mutation is more likely to happen. So it's more likely to be sampled. And then on top of that, we impose, of course, the fact of, mutations have to be adaptive to be accepted.
Okay, fine. Thanks.
Okay. So now what's happening is, Pratyush, question?
Yeah, Deepa. So I just had a thought over here, which is on the previous slide, the image that you showed to explain how the model works in simulating the adaptive work. So I was just wondering, based on what Susanna told us, if we assume that the fitness landscape is not smooth, but is rather like a single step mutation can take us, it need not take us in the neighborhood of fitness, but it can take us to, say, a jump, like it can result in a jump to a very high or very low fitness level. So if we try to incorporate that over here, do you think that would change anything in the way the model, like in what the model tells us?
Yeah, so Lindy did try to do that. So she increased the ruggedness of the landscape, for example, right? So here it's a very simple cartoon. There is one peak in this, or maybe another one there, but it's not accessible in some sense, right? And a single step mutation is only going to take you over here. But if you increase the ruggedness in the landscape by, for example, increasing the epistatic interactions between different genotypes, it's possible that, again, just as a cartoon, I could start from this position and then one mutational step is actually taking me here. Right? So you're traversing a whole fitness valley with that one single step. Is that making sense?
Right. Yeah. It is.
So she tried asking in the simulations, if I change that ruggedness, do the outcomes change? And they don't. They largely remain consistent.
Okay. Yeah.
So of course, it will change the rate of adaptation. It will overall, right, in the adaptive walk, how rugged your landscape is, and so on. So that changes. But the differences and the impact of changing the mutation bias, as you'll see, it is not dependent on that. I won't show you the results for that, but it's there in the bioarchive paper if you want to look.
Yeah. Okay. Thanks.
Any other questions about the walks so far? Okay, good. So we do a bunch of these walks, and I'm just going to show you a cartoon again of, not a cartoon actually, it's real outcomes of what happens when we do these walks in this, right? So your average fitness over time increases as it's expected to increase in a walk, and the rate of that increase kind of saturates at some point, or comes to saturation, close to saturation. And the fraction of beneficial mutations available to your population is also going to reduce over time, right? And so this is something that you have hopefully seen in multiple talks by now, that this is the normal expectation of how an adaptive walk proceeds, all right? So our walk proceeds like this. And so as I mentioned, at different points during this adaptive walk, we're going to estimate the DFE by using the sampling of single step mutations around a particular place where the population is. And we're going to calculate the fraction of beneficial mutations available to the population at each step along this mutational to this adaptive walk, all right? And then we're going to ask, this DFE fraction is effectively what we measured, right, in our experiments originally. And so we're going to ask, can we explain the difference in fraction of beneficial mutations that we observe in our mutY versus wild type strain in a more generic framework like this? So we're going to subtract the fraction of beneficial mutations observed in these DFEs, in these simulated DFEs or possible DFEs available to the different populations, given that the existing mutation bias is that mimicking mutY versus it is mimicking wild type. Okay, so that's what I'm going to plot on the y-axis here. So any values in the top half of this plot indicate that a bias shift is beneficial, and bottom half indicate values where bias shift is deleterious. On the x-axis, I'm showing you what is the transversion bias that we are simulating in a given simulation. So a no bias situation, to help orient you, is going to be roughly here, about 0.6, 0.7. The wild type bias sits here, okay? And the mutY bias is the extreme transversions of 95% that you saw earlier, okay? So we're effectively asking, starting from a population and ancestor, which is like the wild type, which samples more transversions than transitions. If we now simulate a shift to a mutY-like bias, what is the outcome in terms of the proportion of beneficial mutations available to that population, okay? And so this is a bit complicated, but let me walk you through it. What we're doing is we're asking now different kinds of time points during the adaptive walk when we simulate this shift, okay? And why we do that is it's a good way to ask, depending on where the population is in that landscape at that point, which means how long it has adapted already with the wild type mutation bias, it's going to have different fraction of beneficial mutations available to it. You saw this in the previous plot, right? And this is a good proxy for saying how adapted or how well adapted is this population in this landscape, right? So if it has a lot of beneficial mutations available to it, it means that it has not evolved in this landscape for too long. Okay, so it's early on in the adaptive walk that we're now suddenly asking it to shift to a different bias. In contrast, when it's 4%, so these light colors here, it means that it has adapted in this landscape for a long time, very few beneficial mutations available to it. And then we're asking what happens if we now suddenly shift. So what happens is, by and large, you see that anytime you move from a wild type bias towards the opposite side, so you're crossing this no bias line shown in pink, right, towards the mutY, you're always in the regime where bias shift is beneficial, no matter where your wild type population is in the landscape at that point of we are simulating that shift. In contrast, anytime you are going from the wild type bias and reinforcing that existing bias, which means you make it even more biased in the same direction, you make it have even more transitions, it is always deleterious, no matter where your wild type fitness is, how long it has adapted in that landscape. Okay, so what that means is a bias, as when you reverse the mutation bias that exists in your evolved population in that landscape, you're always going to have a benefit when you reverse that bias. Yeah, you were transition biased earlier, you go to transversions, you'll always have a benefit. And another important outcome from these simulations is that magnitude of the benefit is dependent on how many beneficial mutations did you have available to you. In other words, how well adapted you were. So if you're early on in the adaptive walk, you've only recently started adapting in this landscape, you have a relatively small benefit. So these dark lines here, okay, to be a small gain from the predicted gain from the jump to a different reversal of your bias. Whereas if you've been in this landscape for a long time, you accumulated lots of beneficial mutations already, you now have very few beneficial mutations available to you, then you're going to see a huge benefit of changing your mutation bias in the opposite direction. Now, what is going on here? Basically, what we're doing is, like Nikhil was saying earlier, when we change the mutation bias, we're jumping to a different point in the adaptive landscape, in the fitness landscape, right? Where we now have access to a whole set of mutations, which the ancestor was simply not accessing at a high rate, right? So because our wild type was always sampling more transitions than transversions, any beneficial transversions that existed in this bigger landscape, it was simply not sampling them at a high enough rate, which means all the beneficial mutations that are accessible through transitions, most of those it has already sampled in its millions of years of evolutionary history, right, with that same bias. But now, if we suddenly shift it to a transversion enriched bias, what happens is it now has access to transversions. Now, of course, not all transversions are beneficial. They're also deleterious, right? But the beneficial transversions have not been sampled before. And that's the key point. And so now it has access to beneficial transversions. And therefore, our mutY strain shows a DFE which is more skewed towards transversions being beneficial. Right? And so the upshot of this then is that the reason our mutY strain is better or should be better at adapting, that's a prediction from our DFE so far, is not because transversions per se are beneficial. Transversions will not always be beneficial. It's simply that its ancestor has not sampled enough transversions. If now imagine you give me another species of bacterium which has already a transversion bias, I will predict that now if I make a mutation in that strain, which causes it to have a transition bias, that should be beneficial for that strain. Okay, so basically what we're saying then is reversals in mutation bias are beneficial. It doesn't matter what type of mutation it is. And this is great because people have been observing these transversion biases for a long time. These biases, these types of mutation biases exist for other kinds of mutation classification as well. And we have never had any good answer to say why those biases exist. Why do we see that in some cases transitions are better, in some cases transversions are better? Okay, and now we have one sort of generalized way to explain these kinds of effects that we've seen. Okay, so we can use the same simulations now to also ask the question of what about an unbiased ancestor, right? Maybe the intuitive thought is that, well, if you're an unbiased ancestor, then isn't it the best because then you sample all kinds of mutations equally, right? And actually, that's not the case. So we do the same kind of simulation starting with an unbiased ancestor, right? So 0.66 proportion or probability of transversions. And what you find is that shifting from that unbiased ancestor to any other even extreme bias actually has no consistent selective advantage or disadvantage. So it's a neutral shift. Okay, what that means is anytime you move from an unbiased ancestor to a biased situation, there is no selective advantage or disadvantage. There's nothing pushing you back towards the no bias situation, right? But if you can move from an unbiased situation to a biased situation, let's say over here, okay, you're not particularly better off or worse off. Then you keep evolving with that bias neutrally for some time. But now if you evolve with that bias for long enough, you're going to be back in the regime we were in before, where now, from this side to this side, you're going to be benefited because for a while in this landscape, you did not sample transversions. Okay. So a no bias state is not stable. It is not the most stable solution and can easily drift to a bias state. Okay. And by drift, I mean, of course, the population genetic drift, but from a practical perspective, it is also easy because we know a lot of cases. This is very well studied in bacteria in particular, where if we change or if we delete or there's a loss of function in DNA repair genes, okay, such as the mutY gene, which is one example, you end up causing a sudden shift in the mutation bias quite a lot. And many different enzymes are known to have this effect. And people have shown already in the past that there's a lot of these loss of function mutants floating around even in natural populations of E. coli or Salmonella, right? So what that means is there's already existing a lot of polymorphism for mutation bias in natural populations. And we know that it is easy to generate a mutation bias or change a mutation bias because loss of function mutations are relatively easy to for them to occur in natural populations. And so once a mutation bias arises, it can evolve under selection. Pratyush?
Amir has a question in the chat. I wasn't sure if you're able to see. So,
Amir, can you please?
Yeah, I think he's unable to unmute himself and ask. So maybe I can read it out. He's just asking, are transversions more likely to have more positive and larger fitness effects in the model?
In the model, no, there is no, the fitness effects are not encoded in the model in a different way at all, right? The fitness effects of a single step arise from the underlying fitness landscape. So some of them will be large effects, some of them will be small effect, but there's no, that's not coded in the model. Okay. All right, so then we asked, in real lineages of bacteria, do shifts in mutation bias occur often during long evolutionary timescales? And remember, the prediction from the simulations is that we should see reversals of mutation bias very often and a reinforcement of the bias very rarely. Okay, so we asked, to address this, we mapped evolutionary changes in bias in bacterial phylogeny. So I'm going to show you a cartoon image of what we did exactly. So let's imagine this is a phylogeny of bacteria. The real phylogeny has thousands of species in it. And we are able to use homologous modeling to basically say, given that we know the sequences of DNA repair enzymes for a bunch of well-studied bacteria like E. coli and others, Bacillus and so on, we search for homologous sequences in the genomes of other bacteria. And we say, okay, based on that, we can map the presence absence of different DNA repair enzymes, right? So let's imagine there are 3 enzymes here, and we say, okay, different bacterial taxa have these enzymes present or absent. Then what we do is we use data from knockouts, which are gene knockouts known in E. coli, for example, to say, okay, we know that in E. coli, if we knock out the yellow gene, we end up getting a high transversion bias. If we knock out the red gene, we end up getting a high transition bias. Okay, let's imagine that we know this for E. coli. We assume that something along similar lines is going to happen in all the other bacteria. And there are some data suggesting that the direction of these bias changes is the same across different bacterial taxa for which we have data already. So this is not a terrible assumption. The magnitude of that change, of course, will differ across bacteria. But again, that magnitude in cases where we have data does not change too much. So this is a reasonable assumption to start with. Based on this, we can infer bias of other taxa, given the fact of us knowing what enzymes they have or don't have, okay? And then we can say, all right, so this is the predicted transition transversion bias of these other taxa, given what we know about presence absence of the enzymes. We can then do what's called ancestral reconstruction to say, okay, what was the ancestor like? What was its ancestral bias? We map enzyme gain and loss on the phylogeny using, again, ancestral reconstruction methods. So for example, here the yellow gene is predicted to have been lost in this lineage, but was present in the common ancestor of this lineage, all right? And then finally, we can say, based on this, we can ask what is the likelihood of there being, a change in the enzyme status or a change in the mutation bias in that lineage, okay? So here, for example, we would say there has been no change in the mutational bias from the ancestral state. Oh God, I went back again. All right. And similarly, we can say, what is the situation? How many times in which lineages do we predict that there has been a single change in the mutation bias, where as a result of the loss of this enzyme in this lineage, we predict that the mutation bias will have changed from a slightly more transition skewed bias to a more transversion skewed bias, and so on. Okay. And then, of course, the cases that we're most interested in are cases where the bias has been reinforced or reversed, where the dark blue, for example, gets to be more dark, or the dark blue becomes more gray or lighter. Okay, then we do this exercise for an actual phylogeny of bacteria. So here's part of it that I'm showing you here. And each circle corresponds to, each colored circle corresponds to one particular DNA repair enzyme, which we have mapped, for which we've mapped the presence absence shown here in the gray bars. So gray means present and white means absent. And you can see that some DNA repair genes are consistently absent in entire groups of bacteria, whereas there are a whole bunch of them which are generally present in most bacteria. Based on the process that I just described to you, we can then predict what is the expected or estimated transversion or GC280 bias here. So ignore the GC280. I didn't have time to talk about this today, but focus on the transversion transition bias. Does somebody have a question? Okay. And so then based on this whole series of assumptions and estimations, what we can say is, how often do we find that the ancestral, we predict that in this phylogeny, the ancestral mutation bias has been reversed versus reinforced? What we see is that about 82% lineages show at least one reversal in the transversion bias. And a null expectation where we scramble the presence, absence, or gain loss of enzymes in the phylogeny gives us a value of 72%, right? So compared to the null, based on the topology and events and so on, what we're saying is reversals of bias are more enriched, and reinforcements, on the other hand, are very, very rare in general. So they occur only 12% of the lineages, right? So as predicted by the simulations, the reversals of mutation bias seem to occur more frequently and very often in bacterial lineages. Okay. So then to summarize, what we're saying is there are shifts in mutation bias that can open new adaptive paths. So for example, earlier you were only sampling this area of the landscape. And now if you have a shift in the mutational bias because you've shifted to a different position in the transversion ratio, for example, you can access other fitness peaks which were not available to the ancestor earlier. So selection favors a change in bias, and the more extreme the shift, the better it is going to be. Question, Pooja?
Deepa, that was very cool. I just wanted to ask you if, so you took the phylogeny ancestral state reconstruction approach to see if reversals were found in the course of evolution, but do you think we could also look forward and take an experimental evolution approach, or are the timescales very off?
Yeah, so timescales are difficult, but I'll come to this actually in a minute. But yes, that's exactly where we should be going, and we are going there. Okay, so the prediction then is that bias reversals are generally beneficial and evolutionary history is key. We don't need to invoke specific selection to explain bias, for or against a particular type of mutation. We're saying that it depends. Whether a particular bias will be beneficial or not depends on the evolutionary history of that lineage. We also predict that when we alter the rate and the basis of evolution, when we alter the mutation bias, we're going to change the rate and the basis of evolution. We predict that this should reduce the rate at which Muller's ratchet will progress. It should also increase the rate of adaptation, right? So this is kind of the forward stuff that you're just talking about, Pooja. We also predict that mutation rate and spectrum should jointly determine the dynamics of populations. So I think you've heard about mutator lineages already in this course before, where many times what happens is when we delete some of these DNA repair enzymes, we end up increasing or decreasing the rate of mutations as well, okay? Because these are DNA repair enzymes after all, and we're inhibiting their function, right? So when we delete the mutY strain, mutY gene in our case, we also end up increasing the mutation rate. Now, what we did in our experiments is we removed that effect by only looking at single mutations, right? But of course, if we look at MA lines for longer periods of time, we are going to observe that the wild type Muller's ratchet, for instance, is going to progress at a different rate, or should progress at a different rate compared to the mutY Muller's ratchet, because the mutation rate is different. What we're saying is on top of the mutation rate, there should be an additional effect of the bias effect itself, because the DFE is different, right? So we're now trying to combine both of these effects in models and simulations, which Lindy is doing, of course. to try and ask, can we make precise predictions of how this process should turn up, both in the case of evolution under drift as well as evolution under selection. And so then to address the selection question, what we've done is, as you pointed out, Pooja, time scale is a problem, right? Because these are things that are going to happen at very long time scales, typically, not at very short time scales. So we asked if Richard Lenski would share his lines of E. coli, which have been evolving in his lab for 70,000 generations. And so that's a time scale we can work with. Already in their experiments, they have observed the rise and fall of mutator lineages, right? And all of that is predicted based on mutation rate differences. But we think we should be able to also test some of our predictions. So what we expect is from the Lenski lines, if we take populations that have now adapted for 70,000 generations, and we make a mutation bias shift in those populations, we expect it should be much more impactful to change the mutation bias in those populations compared to the same lineages at an early stage of adaptation. So let's say 1,000 generations of adaptation, where they were still able to access a lot of beneficial mutations in that landscape with their existing bias, right? So there we expect that changing the bias should not make a big difference. There will, of course, be an impact of changing the rate of the mutation at that early stage, which, like I said, has been observed already in those populations. So we're hoping to get these lineages in our lab soon, and then start making these simple mutations, which will change the bias, and test some of these key predictions we make. We're also testing some of these predictions using competition, head-to-head competition experiments in the lab in short term. This is work that Pratyush is doing in the lab, and he has a poster about it. If you haven't seen it, please do go talk to him about it to see what he's found. All right, so I'll end by thanking everybody who's been absolutely important, and without whom this work would not be there at all. So the biggest person to highlight is Mridula. She was a PhD student in the lab earlier, and a veteran of this PubGen course. I think she's maybe the only person who's attended all of them, all of the 5 of them, apart from me and Kavita. And so she's back in the lab for some time as a postdoc and continues exploring these ideas that developed as part of her thesis. Bhumika, who's also here, was an undergraduate intern a couple of years ago in the lab, and she worked with Mridula to collect a bunch of the data that I showed you today. Gaurav was a former PhD student. He did all the phylogenetic mapping for the enzyme work that I mentioned to you. And Lindy, of course, is our collaborator who's doing all of the simulations and the mathematics that goes behind understanding all of these things. And I'll also mention all the funding sources and our NGS facility at NCBS, without whom we would not be able to sequence the massive amounts of stuff that we had to sequence. Okay, I think I have maybe a few minutes left for questions, so I'll take questions. Yeah, I can't see who's raised their hands, but.
Deepa, can I ask a question?
Yeah, please.
I didn't understand when you said a slower Mueller's ratchet leads to faster adaptation. How exactly does that happen?
No, those were 2 separate things. Sorry, I should probably have made points. So one prediction is that Mueller's ratchet should be slower if you have this reversal of mutation bias. And the second prediction is that adaptation under selection should be faster if you have a reversal of bias. So basically, to give an example, with the mutY situation, what we expect is if without a shift in mutation bias, but with the same rate, if you expect that this is the rate at which the ratchet would click, with the mutY change in bias, we expect that slowing down should be a little bit shallower, right? And conversely, during adaptation, we expect that if the mutation rate benefit of having a higher mutation rate is, let's say, allowing the population to go this fast, with the bias shift itself, it should go a little bit faster than that. Yeah, so that's the prediction.
All right, thank you.
Hi, Deepa. Thanks for a wonderful talk. I was wondering that with a mutY, I mean, the difference in the mutation rate, do you have to look for scan different number of lines to get similar number of single mutations?
Yes. Yeah.
Okay. So how much is the difference ballpark? I mean, could you just say
it depends on the mutation rate, right? So we've done similar. I mean, we haven't done. We're in the process of doing similar experiments for other mutators, and how many we have to scan, and how. So we can also alter the length of the MA experiments, right? So for the wild type, for example, we had to do, Mridula had to do 3 months of MA lines to get to the point where we got these 80 with single mutations. But with the mutY, just a few days is enough. So.
And another question I had, that I remember in this graph you showed that if you look at actually the wild type and a mutY in different media, let's say with the different carbon sources, their baseline fitness is not very different, right? They more or less do similarly. In that case, can you actually determine the DFEs, let's say with one common ancestor? Would that help your purpose? Like, while you're thinking of DFEs, maybe a third culture altogether, which is similar, or maybe just one of them, would that change anything? What do you think? Like,
well, it's hard to define DFEs unless you use the same ancestor, though, right? Or the appropriate ancestors. So DFE is defined based on some random third ancestor would just not make sense at all in my mind.
Okay, thanks.
Somebody else has more questions?
Yeah, thanks for the talk. I think it's still not very clear to me what mutational bias means in the, like, how do you measure it in the experimental setup?
So, okay, so let's imagine with these MA lines, we have 100 mutants that we got, okay,
Yes.
of the wild type and the mutY strain. We just count up what fraction of those mutations are transitions and what fraction are transversions. And
Okay.
that ratio is dramatically different, so the mutation bias is different.
Okay, yeah, okay, now, yeah.
It's fairly simple, yeah.
And it's the same,
Sorry.
yeah.
Okay. Maybe I should stop sharing. I don't know if there are questions in the chat that I should look at. Okay. Okay, I don't see any other questions. So thank you, everybody. And yeah, there's a lot of really fun stuff that comes out of this predictions, and I'm so excited about this. This might be the most interesting things. And I must say that, I have to thank the ICTS PubGen school, because without it, I would not have, spent all this time with Lindy and talked, and we passed notes in some of the lectures, but it was fun. So I'm really excited. Thank you, everybody. And so we'll be back for our last session of posters at 4:30. So please be there, and see you all there. Bye.